Welcome to the Deep Dive, where we take complex information and cut through the noise to give you the most important insights. Today, we're plunging into a topic that touches so many lives: Alzheimer's disease.
hmm
It's a condition that presents enormous challenges, particularly when it comes to the sticky protein deposits found in the brain, the beta-amyloid plaques.
That's right. These plaques have long been, well, considered a major factor in Alzheimer's progression. And tackling them effectively, ideally in a way that isn't highly invasive, that's a huge goal for researchers worldwide.
And that's precisely what caught our attention in the source material for this deep dive. We're going to explore a cutting-edge scientific study, really fascinating stuff, that investigates a novel, non-invasive approach.
Using electric fields.
Exactly, using electric fields specifically, oriented external electric fields with the potential to actually break down these incredibly stubborn beta-amyloid plaques.
Our source is a fascinating paper published just last year in the prestigious Journal of the American Chemical Society. It's by a team including Kalita, Bergman, Dubey, and Shake.
Okay.
And importantly, this isn't an experimental study on patients or animals, not yet anyway. It's a deep dive right into the molecular world using these powerful computational techniques called molecular dynamics simulations.
Right, simulations. Sounds complex, maybe a little intimidating, but our mission today is really to distill the absolute key findings from this paper.
Yeah, break it down.
Break it down exactly, help you understand what these researchers did, what they discovered about fighting these plaques right at the atomic level, and, what it could potentially mean for the future.
Okay, let's unpack this. To really appreciate the study, I think we first need a little bit of background on Alzheimer's itself, and specifically these beta-amyloid plaques.
Absolutely. So Alzheimer's disease, it's incredibly common, isn't it? It's this progressive neurodegenerative condition that, well, it slowly erodes memory, thinking abilities, behavior.
Yeah. And sadly, it disproportionately affects older individuals. Its global footprint is just staggering. It's estimated to account for about half, maybe even 60% or so, of the nearly 50 million people living with dementia around the world.
50 million, wow.
Yeah. So the need for effective treatments, something that can slow it or halt it or even reverse its progression, it's just immense.
And for a long time, the dominant scientific explanation for why Alzheimer's develops has centered on what's called the amyloid cascade hypothesis.
Right, the amyloid cascade. This hypothesis basically proposes that the main driver of AD is the buildup and clumping together the aggregation of a specific peptide called beta-amyloid, or ET.
Ah, got it.
These clumps form outside brain cells, creating those characteristic senile plaques you hear about. So logically, a major focus of therapeutic strategies has been on trying to stop this aggregation or, clear the plaques once they've formed.
Now, it's important to acknowledge science is always a journey, right? There are evolving perspectives, like some research in other neurodegenerative diseases, Parkinson's, for example. It suggests that the problem might not just be the accumulation of these misfolded proteins, but perhaps also a loss of the normally folded functional protein.
True. The field is always exploring nuance, looking at different angles. However, the amyloid plaque hypothesis, it remains a very prominent framework in AD research.
Still central.
Very much so. And finding ways to decompose these plaques is still a major goal. And critically, if breaking down the plaques could somehow help restore or, free up the properly folded, healthy forms of the protein, then tackling the plaques remains a vital therapeutic target.
Okay, so let's focus on this beta-amyloid peptide itself. Abby, where does it actually come from?
It's actually a piece that gets cleaved off, kind of snipped from a larger protein that's embedded in the cell membrane. That larger one's called the amyloid precursor protein, or APP. Now, the cell makes different lengths of the A peptide, but one variant in particular, A42, meaning it has 42 amino acids. That one's really significant because it's highly prone to aggregation. It forms these small clusters first.
Your little seeds.
Kind of, yeah. They're called oligomers. And some researchers believe these oligomers are particularly toxic, maybe even more so than the big plaques themselves. They're also being studied as potential early diagnostic markers. Then these oligomers build up into the larger fibrils and eventually the plaques.
Okay, so studying the full-length F42 peptide in detail, seeing exactly how it aggregates at the atomic level, that sounds like a massive computational challenge.
Oh, it really is. Simulating the complex behavior of a whole protein segment like AR42 over, meaningful timescales. It requires immense computational power, just huge amounts.
Right.
And this is where the researchers in this study made a really smart move for their computational experiment. Instead of trying to simulate the full-length peptide, they focused on a smaller, much more manageable piece.
A shortcut, kind of.
A representative shortcut, yes. A 7-residue segment with the specific sequence N-acetyl, KLVFAE-NH2. This little piece corresponds to residues 16 through 22 within the full F peptide.
Okay, KLVFFAE. Why did they pick that specific tiny snippet? 7 amino acids seems pretty small.
It seems small, but there's a really good reason. Back in the year 2000, a key experimental study by Tico and colleagues identified this precise 16 to 22 segment as the smallest portion of the Takeda peptide that could still, on its own, form ordered fibrils, the structural basis of the plaques.
Ah, so it retains that key plaque-forming behavior.
Exactly. As they show in Scheme 1 of the paper, this small chain is able to aggregate into what are called anti-parallel beta sheets and form these ordered fibrils when it's just sitting in water.
Anti-parallel beta sheets. We'll probably come back to that term.
We will.
Yeah.
But the point is, this behavior closely mimics the initial steps of how the full-length peptide forms plaques. So it serves as a really effective computational model. It lets them study the fundamental physics of aggregation and, importantly, decomposition without needing to simulate the full, much more complex molecule.
It's like a perfect representative building block for their computer model. Very clever.
Precisely. Understanding the detailed molecular structure and how these fibrils form, starting from maybe less stable early forms called metastable protofibrils, building up to the mature fibrils, it's absolutely crucial for developing strategies to, well, ameliorate AD.
Right.
And researchers use all sorts of techniques for this, experimental ones, of course, but also sophisticated computational ones, like the molecular dynamics simulations in this paper. They let you peer into these processes at the atomic scale. And this study focuses specifically on the mechanism of plaque formation and, critically, plaque decomposition.
Which brings us right to the core of their approach, classical molecular dynamics simulations, often called MD. How did they actually set up these computational experiments? What does that involve?
Well, at its heart, molecular dynamics simulates how atoms and molecules move and interact over time. It does this based on, the fundamental laws of physics and the forces between atoms. Think of it like creating a virtual box of molecules.
Okay.
And running a really sophisticated physics engine to see what happens second by second, or rather femtosecond by femtosecond.
All right, so what was in their virtual box for this study?
They started with 10 of those small 7-residue peptide chains, the KLVFFAE ones. These were initially placed randomly, sort of scattered and relatively far apart, within a box filled with water molecules.
Just floating around?
Yep. They used a common model for water called TIP3P. It's known to accurately capture many of water's properties that are important for these kinds of simulations. They set the concentration of peptides to 0.054 molar. Now, that's quite high within the small simulation box.
Higher than in the brain, probably.
Oh, almost certainly. But it's chosen deliberately for the simulation. It makes sure the peptides are likely to bump into each other and interact and potentially aggregate within the simulated time frame, which is computationally limited. And the overall system was kept electrically neutral.
Okay, so you've got the peptides and the water in the box, and the simulation just begins.
Well, first, there's some careful preparation. They perform energy minimization steps. This is basically to relax the system, resolve any unrealistic starting configurations, like making sure no atoms are crashing into each other in an impossible way.
Tidying up the starting point.
Exactly. Then they gradually heated the system computationally up to 300 Kelvin. That's roughly human body temperature.
Okay.
This is followed by an equilibration phase. The system is allowed to settle down at 300 K in standard atmospheric pressure, making sure it's in a stable representative state before they start the main observation period. Then came the production runs, where they actually simulated the dynamics and watched what happened.
And how long did these production runs last in the simulation? You mentioned limited time.
Right. These simulations ran for durations ranging from 500 nanoseconds up to 1200 nanoseconds. That's 1.2 microseconds.
Okay.
And the key ones, especially looking at what happens after applying a field, went even longer, up to 1.5 microseconds.
Nanoseconds, microseconds. These are incredibly short timescales in our everyday experience.
They are. A nanosecond is a billionth of a second, a microsecond, a millionth. Tiny, tiny windows of time.
Unimaginably small.
Yes, but for the movement and folding of molecules, these timescales are actually where a lot of the important action happens. Microsecond scale dynamics can reveal crucial behavior, like proteins folding, or in this case, aggregating.
Right.
And the authors make a point of noting that while the simulated time is, say, 1 microsecond, The actual computation time required to run these simulations on powerful supercomputers could take weeks.
Weeks of real time to watch something for a millionth of a second. That really highlights the complexity, doesn't it? Simulating all those atoms interacting.
It really does. The sheer number of calculations is immense. And to make sure the results weren't just a fluke, like maybe happening only because of the specific way they initially placed the peptides.
Yeah, how do they avoid that?
They use what's called the multi-trajectory approach. This involves running the simulation multiple times, starting each run with slightly different random initial conditions or, say, different initial velocities for the atoms. It allows the system to explore a wider variety of possible pathways and configurations.
So it gives more confidence that the behavior they see is general, not just an accident of one particular simulation run.
Exactly, more robust results. And they had two main sets of these simulations, correct?
Yes, broadly speaking. One set was the control group. This simulated the peptide behavior without any external influence, just the peptides in water doing their thing, no electric field applied.
The baseline.
The baseline. The other set involved applying these oriented external electric fields, or OEEFs. They tested different field strengths, and importantly, they also tested oscillating fields at different frequencies, which, as we'll see, turned out to be absolutely critical.
Okay, OEEFs, static and oscillating. Got it. And were there a lot of other sort of technical complexities they had to handle in the simulation setup? You mentioned TIP3P water.
Oh.
Yes. Like any rigorous simulation, they used a whole suite of standard advanced techniques to ensure accuracy and realism. Things like methods to control the pressure. They mention Monte Carlo barostat. Running multiple copies of simulations called replica simulations, just to check for consistency.
Right.
Using algorithms to handle specific molecular constraints like keeping bond lengths fixed. They mentioned the shake algorithm. Employing sophisticated methods for calculating the long-range electrostatic interactions accurately. That's where things like particle mesh, Ewald, or PME come in.
Without me, okay.
And using periodic boundary conditions, PBC. This is a clever trick. You simulate one box, but mathematically, you treat it as if it's surrounded by infinite identical copies of itself, like tiles on a floor.
So a peptide leaving one side of the box just re-enters from the opposite side.
Pretty much. It avoids weird edge effects and helps simulate a larger, more bulk-like system, even with a computationally manageable box size.
So, okay, well, we don't need to get lost in the weeds of every single acronym. The takeaway is they used rigorous, well-established methods. They tried to make their simulation as accurate a representation of the physical system as possible within the limits of the model.
Precisely. The overall goal was clear: to systematically study beta-amyloid aggregation and, crucially, its disruption under both static and oscillating OEEFs. They wanted to really describe at a molecular level.
Oh, what happens?
Yes. How aggregation happens spontaneously. Then see if OEEF could prevent it if you start from random chains. Then investigate if OEF could break down preformed aggregates. And then, really importantly, compare their findings, especially with the oscillating fields, to the characteristics of existing therapies like deep brain stimulation, DBS, and TT fields.
To see if it offers advantages.
Exactly. They aim to provide a solid computational basis, a rationale, for potentially using electric fields as a therapeutic strategy down the line.
Okay, fantastic setup. Let's start with that baseline then, the control group. How did these plaques actually form spontaneously in their simulation when there's no external electric field pushing or pulling on the molecules? What drives it?
What's truly fascinating here and center of the whole story is the intrinsic nature of these small peptide chains. If you look back at scheme 1 in the paper, it shows the chemical structure. Each individual N-acetyl KLVFAE NH2 chain has a very large inherent dipole moment.
Right, a dipole moment. We touched on this. It's like having a positive charge concentrated at one end of the molecule and a negative charge at the other, creating a separation of charge, like a tiny magnet.
That's a good analogy, yeah. In this specific peptide, this charge separation arises naturally from the chemistry at the ends of the chain, the acetylated N-terminus and the amidated C-terminus. And the resulting dipole moment is quite large. The paper quotes it around 125 dB. That's a significant dipole for a small molecule.
125 dB, okay. And molecules with large dipole moments, they tend to interact strongly with each other, don't they?
They do. They experience mutual attraction due to these dipole interactions. It's like those tiny magnets wanting to line up or attract each other, positive end to negative end.
So these dipole forces are pulling the peptides together. Is that the only force really driving the aggregation?
No, it's not the only one. Hydrogen bonds are also incredibly important. These are specific relatively strong interactions that form between the backbone atoms of different peptide chains.
Like a molecular glue.
Kind of, yeah. Specifically, they form between a nitrogen atom and its attached hydrogen on one chain's backbone and an oxygen atom on another chain's backbone. When the chains line up correctly, they can form a whole network of these hydrogen bonds, which really locks the structure together.
So you've got the longer range magnetic like dipole attraction pulling them close, and then the shorter range hydrogen bonds kind of gluing them together once they're aligned.
That's a great way to picture it. Dipole brings them in. Hydrogen bonds stabilize the specific structure.
Okay, so how did the simulation actually show this process unfolding over time without any external field?
The results of the control simulation are visually quite striking. They show snapshots in Figure 1. At the very beginning, time 0 nanoseconds, the 10 peptide chains are just random, disordered coils. They're floating relatively far apart from each other in the water box. No defined secondary structure at all.
Just scattered like molecular spaghetti.
Exactly. But as the simulation time advances, they show snapshots at 250 ans, 500 ans, and 1,000 ans, which is 1 microsecond. You clearly see the chains starting to move towards each other.
Pulled by those dipoles.
Pulled by the dipoles, yes. And as they come into proximity, driven by those forces and the potential to form those hydrogen bonds, they begin to fold and arrange themselves into ordered structures.
And these structures are the antiparallel beta sheets we mentioned.
Yes. They transition from being random coils into these specific beta sheet configurations. And critically, not just any beta sheets, but specifically antiparallel ones. This means that adjacent peptide strands in the sheet run in opposite directions, head to tail, essentially.
The antiparallel. Okay.
These antiparallel sheets then pack together in a very specific way, forming structures that the paper describes as cross beta sheets, or having a hairpin-like arrangement. And the authors point out that these specific structural motifs are known starting configurations, reported experimentally, for the formation of the larger senile plaques found in AD brains.
So the simulation spontaneously reproduces the core structural units thought to initiate plaque formation. That's pretty convincing for the model.
It really is. And these initial aggregates, these packed antiparallel sheets, are held together and stabilized by both forces we discussed. The interchain dipole interactions and the network of hydrogen bonds formed within and between the beta sheets.
And they have data to back this up.
Oh yes. Figure 2 provides quantitative evidence. Figure 2a gives a nice illustration showing how the hydrogen bonds form between those antiparallel strands. They even label in bonds ones already formed within the sheet and out bonds, which are edges available to interact with other peptides, allowing the structure to potentially grow larger.
Right, showing how the plaque could build up.
And Figure 2b confirms this quantitatively. It plots the number of hydrogen bonds over the simulation time. You see a clear, significant increase as the simulation progresses, directly demonstrating the formation and stabilization of these beta sheet structures. More H bonds means more stable sheet.
Okay, hydrogen bonds confirmed. Now, let's circle back to the dipole interaction. You said the antiparallel arrangement forms. Why is that specific arrangement so stable from a dipole perspective?
This is absolutely key to understanding the spontaneous formation. Remember, each individual peptide has that large dipole moment, around 125 DeV. Now imagine if the peptides lined up in a parallel fashion, all pointing in the same direction, head to head.
Okay.
If they did that, their individual dipoles would mostly add up. You'd end up with a huge total dipole moment for the entire aggregate. This would create a very strong overall electric field around the aggregate, which is generally an energetically unfavorable state, unless there's something external, like an applied field, to stabilize it.
Right. Systems naturally tend towards lower energy states. A giant collective dipole is high energy.
Exactly. The antiparallel arrangement, where adjacent strands point in opposite directions, head to tail, is favored precisely because it allows the individual peptide dipoles in adjacent strands to largely cancel each other out.
They point opposite ways, so the net effect is much smaller.
Precisely. This minimizes the total dipole moment of the aggregate. As they show in Figure 4, the calculated total dipole moment of the 10-peptide antiparallel aggregate formed spontaneously was only about 166 DeV.
166D total compared to maybe 10 times 125D or 1250D if they were all parallel. That's a massive reduction.
It's a dramatic reduction, and this minimizes the aggregate's own electrostatic energy. Figure S2 in the supplemental information visually reinforces this. It shows calculated electrostatic field lines supporting this strong dipole interaction, favoring antiparallel clustering to minimize the overall field.
So the structure forms in a way that reduces the group's collective magnetic field, to use our analogy. It's the most stable arrangement for the peptides interacting with each other.
That's a perfect way to put it.
Huh?
The peptide dipole interactions themselves are most favorable when they align antiparallel. And the stability of this formed aggregate is also evident in the RMSD plot shown in Figure 2C.
Okay, RMSD. Remind us what that tells us again.
RMSD stands for root mean square deviation. It's a measure of how much the structure of the peptides as a group is changing or deviating over time compared to some reference structure, often the starting point or an average structure. High fluctuations in RMSD mean the structure is unstable or changing rapidly. Low stable RMSD means it's settled into a consistent shape.
And what does the RMSD plot show for this spontaneous aggregation?
For the first, say, 300 nanoseconds or so, the RMSD fluctuates quite a bit. That makes sense. That's the period where the peptides are still mostly disordered, moving around randomly, exploring different interactions, figuring out how to come together.
The initial chaos.
The initial chaos, yes. But after about 300 nanoseconds, the RMSD plot flattens out and stabilizes at a relatively low value. This tells us that the system has settled down. It has converged into a stable, defined configuration, which corresponds to that well-formed antiparallel beta sheet aggregation we saw in the snapshots.
It found its happy place, structurally speaking.
It found its minimum energy configuration, yes, the stable aggregate.
Okay, so let's sum up this fundamental process, the baseline. In the absence of any external field, these beta-amyloid fragments, at least this model KLV-FAE segment, spontaneously and efficiently assemble into stable antiparallel beta sheets. These are the fundamental building blocks of senile plaques. And this self-assembly is driven primarily by the strong attractive forces between their large individual dipole moments and the formation of stabilizing hydrogen bonds.
That's the inherent tendency, the default behavior of these peptides in water, according to the simulation. They naturally want to clump together in this specific, ordered, and ultimately harmful way.
Right. Which leads directly, logically, to the next big question for the researchers. If this aggregation is driven so strongly by those dipole interactions, shouldn't applying an external electric field, an OEDF, be able to interfere with that? Maybe even prevent this aggregation from happening in the first place?
That was exactly the key hypothesis they wanted to test. Since the large peptide dipoles are so central to the spontaneous assembly process, an external electric field should, in theory, be able to compete with those internal forces and maybe redirect the peptide's behavior.
Makes sense. Competing forces.
So they ran a new set of simulations. They started with the same random arrangement of peptides in water, but this time, right from the beginning, they applied a static-oriented external electric field.
And what did they find when they turned on this static field? Did it stop the aggregation?
Well, they tested different field strengths, applying the field along the x-axis of their simulation box at a relatively low strength, 0.01 volts per angstrom.
Volts per angstrom. Okay. That's a measure of electric field strength at the molecular scale.
It is. At 0.01 V, they observed that it did slow down the rate of aggregation compared to the no-field simulation. The peptide still eventually aggregated, but it took noticeably longer, pushing into the microsecond scale.
So, low field strength hindered it, put the brakes on a bit, but didn't stop it completely. What about a higher strength? Did they try that?
Yes, they did. They increased the static field strength to 0.02 VO. And here, the effect was much more pronounced. At this strength, the propensity for the beta-amyloid peptide chains to aggregate into that problematic antiparallel structure was significantly reduced.
Really suppressed it.
Significantly suppressed it, yes. Looking at the simulation snapshots in Figure 3, and also Figure S6 in the supplement, the peptides remained in a much less defined state overall. They didn't form those tight, ordered antiparallel sheets nearly as readily.
Interesting. But did they just stay random coils, or did they see some other kind of structure emerge when the field was on?
Ah, yes, and this is a really crucial observation. At 0.02 V, while robust aggregation into antiparallel sheets was suppressed, they found that a different kind of arrangement became dominant, or at least preferred. This was a parallel beta sheet arrangement.
Parallel. So now the strands are lining up head to head.
Exactly. The peptides tended to align side by side, oriented in the same direction along the field. The paper indicates this with arrows in Figure 3 and shows representative structures in Figure S7. This parallel arrangement was clearly preferred over the problematic antiparallel one when the static field was applied.
Okay, that's fascinating and maybe counterintuitive. How does the electric field suddenly make the parallel structure, which wasn't stable before on its own, become the favored one when the field is on? Why the switch from antiparallel to parallel?
It goes back to the interaction energy between the molecule's overall dipole moment and that external field. Remember the formula they mentioned? Roughly A Oki 4.8 minokvex.
Right. The energy change depends on the field strength and the component of the molecule's dipole moment that aligns with the field.
Exactly. The external field applies a force, or rather a torque, that tends to orient the molecule's dipole moment. For maximum energy stabilization, the dipole wants to align in the direction opposite to the field vector.
Okay, so the field is trying to pull or push the peptides to align their individual dipoles in a specific way relative to the field.
Yes. Now let's think about the aggregate dipole moments again, referencing figure 4. In the absence of the field, the antiparallel aggregate was stable because its internal peptide dipole interactions minimized the total dipole moment of the group to that low value, around a 166 dB. That minimized the internal energy of the aggregate itself.
Got it. Low internal energy.
But when you apply a strong external field, a new, potentially much larger energy factor comes into play: the interaction energy of the whole aggregate with the external field. And think about a parallel arrangement of the peptides, where all those big individual dipoles, 125D each, are pointing roughly in the same direction.
That would create a huge collective aggregate dipole moment, wouldn't it?
Exactly. Figure 4 shows the calculation for the parallel arrangement they observed with the OEEF on. The total aggregate dipole moment was about 1131.4 dB.
Wow, okay, 1131 dB. That's nearly 7 times larger than the total dipole moment of the stable antiparallel aggregate, 166D.
It is. And here's the critical insight. This huge parallel aggregate dipole interacts very strongly and very favorably with the external OEEF, especially when it orients itself opposite to the field vector. The paper calculates this stabilizing interaction, and just the interaction with the external field, to be huge, around 108 kilocalmol for the parallel aggregate in the 0.02 V field.
108 kilocalmol. That's a massive energy stabilization just from interacting with the external field.
It's enormous on a molecular scale.
Mm.
And this massive stabilization from the external field effectively overcomes the less favorable peptide interactions within the parallel sheet itself. The parallel arrangement might not be ideal internally, but the external field gives it such a huge energy bonus that it becomes the preferred state overall while the field is on.
Okay, let me see if I get this. The field provides a giant energy reward to the parallel structure because of its huge total dipole moment interacting with the field. Meanwhile, the antiparallel structure, which was stable on its own due to its small total dipole moment, it gets almost no energy benefit from interacting with the external field because its net dipole is so small. One chain interacts favorably, the next unfavorably, canceling out. So, the parallel structure wins when the field is present.
That's it, exactly. The interaction energy with the field is the dominant factor at this field strength, and it strongly favors the high dipole parallel configuration. The anti-parallel structure, which was stable without the field, becomes relatively less stable with the field on because it simply doesn't get that massive stabilizing interaction energy compared to the parallel arrangement.
That makes sense. And they have other data supporting this shift away from aggregation.
Yes, the RMSD plot, again, figure S8 shows the RMSD for the simulation run with the 0.02 V alphatic OEEF applied from the start. And that plot looks very different from the stable aggregation seen in figure 2C. Instead, it looks much more like the very beginning of the zero field simulation. Remember the first 100 nanoseconds or so of figure 2C before aggregation really took hold. High fluctuations, no stable convergence.
So quantitatively, the RMSD shows the system isn't settling into the stable antiparallel aggregate when the field is on. It stays more disordered or transiently parallel.
Precisely. It's strong quantitative evidence that the field is preventing the system from reaching that stable, tightly packed, anti-parallel aggregate state.
Okay, let's recap the prevention aspect then. Applying a static OEEF at a sufficient strength, like 0.02 V in this model, seems to effectively prevent the formation of the detrimental anti-parallel beta sheets. It does this not by making the peptides completely inert, but by favoring a different, less tightly packed structure, a loose assembly of parallel-aligned peptides, which is held together primarily by its strong interaction with the external electric field itself.
Exactly right. It disrupts the natural self-assembly pathway that leads to the stable, harmful plaques.
Okay, so static OEF can prevent aggregation if applied early, but the really big question for potential therapy is, what if the plaques have already formed? Can applying a static OEF also break down existing well-formed aggregations?
This was the next logical and perhaps more clinically relevant step in their investigation. They took a system where the peptides had already been allowed to aggregate into a stable antiparallel beta sheet structure, essentially. They used the final stable state from their no-field simulation run, shown as the starting point in Figure 5a, and then they applied that same static OEEF of 0.02 V to this preformed aggregate.
Okay, so field applied after the plaque model has formed. And what did they observe happening to the aggregate then? Did the field manage to break it apart?
Yes, it did. The simulation showed that applying the static OEES significantly destabilized the preformed aggregate. As you can see in the time evolution snapshots in Figure 5b and 5c, over the course of the simulation with the field on, the chains within the aggregate began to visibly separate from each other. They noted that the aggregates started to noticeably disintegrate around the 500 nanosecond mark.
So it took about half a microsecond to start seeing it break down. And if they kept the static field on longer,
They extended that particular simulation up to 1200 nanoseconds, or 1.2 microseconds. And by that time, the simulation showed the aggregate had completely broken down. The peptide chains were dispersed, reverting back to disordered, random coil structures, with basically no sign left of the original order beta sheet aggregation.
So, conclusion. A moderate static OEEF can, indeed, break down spontaneously formed aggregates in this model. That sounds promising, doesn't it?
It sounds promising initially, but here's this significant drawback, and the authors are very clear about highlighting this. The time it took to break down the aggregate using the static field, roughly 500 to 1200 nanoseconds, was very similar, in fact, almost the same order of magnitude, as the time it took for the spontaneous aggregation to form in the first place, which was around 1 microsecond or 1,000 nanoseconds.
So it takes about as long to break it down with the static field as it does for it to form naturally. It doesn't sound very efficient for a potential treatment, does it? If breakdown is as slow as formation, you're constantly just fighting the natural tendency to reaggregate.
Exactly, that's the problem. The implication for any potential therapy based on this is that to keep the amyloid in a disrupted state using a static field, you would likely need to apply it for very long periods, possibly even continuously.
Right.
And the authors explicitly state that applying a static OEEF for such extended durations is, and I quote, not a good idea for treatment of humans.
Yeah, that makes intuitive sense. Long-term, continuous static electric fields being applied inside the brain. That sounds potentially problematic, maybe harmful in other ways.
It raises significant safety concerns, absolutely.
Yep.
So the bottom line from the static field experiments was, yes, it can prevent aggregation and it can break down existing aggregates, but the breakdown is slow on the same timescale as formation and would likely require continuous, potentially unsafe, long-term application.
Okay. So static fields are maybe not the ideal therapeutic approach despite showing some effect. What were the researchers' options then, based on this finding?
Well, based on this limitation, they considered two main possibilities to potentially improve the efficiency or safety. One option was simply to crank up the static field strength much higher, hoping to speed up the breakdown process.
Just use a bigger hammer?
Essentially, yes. But they rejected this approach, quite reasonably, because significantly increasing the field intensity could easily become harmful to sensitive biological tissues like the brain. More field strength equals more potential for unwanted side effects or damage.
Okay, so stronger static field was out. What was the second option?
The second option, and the one they decided to pursue, was to investigate applying an oscillating OEEF. The idea was to use the same moderate, potentially safer field strength, 0.02 VE, but instead of keeping it constant, make it oscillate back and forth at different frequencies.
Shaking things up instead of just pushing constantly. This brings us to what seems like the core of their most promising finding, the oscillating fields. What was the main goal in exploring these?
The primary goal was really twofold. First, to see if oscillating fields could be more efficient at breaking down the preformed aggregates compared to the slow static field. And second, perhaps more speculatively, but importantly, to identify frequency ranges that might be relevant or potentially safer for use in the human brain down the line.
Okay. And which frequency band did they focus on initially for these oscillating fields?
They started by investigating frequencies in the microwave range, specifically the gigahertz or gigahertz band.
Gigahertz, like Wi-Fi frequencies.
Exactly like Wi-Fi, Bluetooth, cell phones, radar, that sort of range. Their rationale for choosing this range is actually quite interesting and practical. They point out that humans are constantly exposed to microwave frequencies in our everyday environment from all these technologies.
Right, we're bathed in them.
We are. And this suggests that, at certain intensities anyway, fields in this frequency range might be relatively biologically tolerated compared to, say, a much lower or much higher frequencies, where biological interactions might be different or less understood in terms of long-term exposure.
Okay, interesting rationale. So they applied an oscillating OEEF at 0.02 Veloa. The same strength as the static field they found could break things down slowly, but now oscillating, starting with a frequency of zero point 0.1 gigahertz. What was the outcome? Did it work better?
This is where they saw a truly remarkable difference compared to the static field simulations. The results were striking. As shown visually in Figure 6, the disruption of the beta-amyloid aggregate occurred incredibly rapidly with the oscillating field.
Incredibly rapidly. How fast are we talking compared to the static field?
Okay, get this. The oscillating OEEF at just 0.1 gigahertz was able to completely destroy the existing anti-parallel beta sheet pairs and convert the peptides back into disordered random coils in only 50 nanoseconds of simulation time.
50 nanoseconds, compared to the 500 to 1200 nanoseconds it took for the static field to do the same job.
Exactly. 50 nanoseconds versus, let's say, an average of 1,000 Ns for the static field breakdown. That's a factor of 20 faster. And if you compare it to the time it took for the aggregate to form spontaneously in the first place, which was also around 1 microsecond or 1,000 nanoseconds, the oscillating field broke it down roughly 20 times faster than it even formed.
20 times faster breakdown than formation. That is a massive increase in efficiency in disrupting the plaque structure, at least in their simulation model. That's huge.
It's a dramatic difference in kinetics, yes.
Okay, that kind of speed is amazing for breaking something down.
Yeah.
And this feels like the critical question, especially if you're thinking about a potential therapy. What happens after the oscillating field is turned off? Does the broken down aggregate just snap right back together once the shaking stops? Is the effect temporary?
This is arguably the most significant and potentially game-changing finding of the entire study. And it's absolutely crucial for the potential therapeutic relevance of this technique. They designed a specific simulation experiment precisely to test this reversibility question.
How did they test it?
What they did, they took the system after it had been simulated for 500 nanoseconds with the oscillating OEEF applied at 0.1 gigahertz and 0.02 VO. By this point, as we just discussed, the aggregate was completely disintegrated into random coils. Then they simply turned the electric field off completely in the simulation and just continued running it, watching what the peptides would do on their own now.
Okay, field off. And did the peptides start clumping back into those anti-parallel plaques?
No, absolutely not. The simulation converged relatively quickly, structurally speaking, within about 300 nanoseconds after the field was removed. But the key observation, which is clearly illustrated in Figure 7, is that the peptides, now in their random coil states, remain widely separated. They absolutely did not spontaneously return to their original problematic anti-parallel beta sheet aggregation state.
They just stayed dispersed.
They stayed dispersed. Even after 500 nanoseconds after removing the field, the simulation snapshot shows the plaque remained disintegrated. The peptides were still mostly random coils floating apart.
That's incredible. And they tested this over an even longer period, just to be sure.
They did. They extended that specific simulation run, the one where they turned the field off after 500 mans, up to a total of 1.5 microseconds after the field removal. So a very long time in simulation terms. And throughout that entire extended period, the peptides maintained a similar conformation. They stayed in that disordered, disintegrated state.
Wow.
The RMSD plot for this phase, shown in Figure S11, which tracks the overall structural change, remains stable at a high-value characteristic of disorder. It confirmed that the disintegrated state persisted for the full 1.5 microsecond simulation duration after the field was removed.
This seems to demonstrate, at least within the confines and timescales of these simulations, that the disintegration caused by the oscillating OEEF appears to be irreversible, or at least very, very slow to reverse. That is a huge finding, isn't it? Because it implies a potential treatment might only need periodic application, not continuous.
It's incredibly promising from a simulation perspective, yes. That apparent irreversibility, or very long-lasting effect, is perhaps the most exciting outcome. And to see how robust this finding was, the researchers didn't just test that one frequency, 0.1 gigahertz. They tested a range of different frequencies. Figure 8 presents this data quite nicely.
Okay, what frequencies did they check?
They examined 4 different frequencies within the microwave range, increasing by 10-fold increments each time. 0.1 gigahertz, 1.0 gigahertz, 10.0 gigahertz, and 100.0 gigahertz, all tested at the same moderate field strength of 0.02 VE.
So they checked across a really wide spectrum of microwave frequencies, from lower to very high.
Exactly. And in all these cases, they followed the same procedure. Apply the oscillating field for 500 nanoseconds, long enough to break down the aggregate based on the 0.1 gigahertz result, and then remove the field and simulate for another 500 nanoseconds to check carefully for any signs of reaggregation.
And what did they find across all those frequencies? Did the plaques reform?
No. Figure 8 shows the results in terms of the percentage of average parallel beta sheet character detected. Remember, the field tends to favor parallel arrangements loosely.
Right.
Under the oscillating field, this parallel character develops to some small extent. After the field was removed across all 4 frequencies, the percentage of this parallel character increased only very slightly further, reaching a maximum of only about 4% total parallel character on average. This indicates maybe some very minor, perhaps transient local reordering, but absolutely nothing like the formation of a stable, compact aggregate structure.
Okay, so very little parallel structure, and it didn't increase much after the field was off, but the crucial point across all these frequencies was, did the anti-parallel structure come back?
That's the key takeaway. Critically, in none of these simulations, across this entire wide range of microwave frequencies, were there any signs of the problematic anti-parallel beta sheet formation recurring after the field was removed. Zero detection of the harmful structure reforming.
Wow.
This quantitative data strongly supports the idea that the state achieved after the oscillating field treatment is indeed a stable, likely irreversible on these timescales, disintegrated state. The peptides just don't seem to want to go back to being plaques once they've been disrupted this way.
That raises a really important question then. Why? What is it about the oscillating field or the resulting disordered state that prevents the peptides from simply relaxing back into their previously stable, energetically favorable, anti-parallel aggregate form once the external field is gone? What's the mechanism behind this apparent irreversibility?
That's a great question, and the paper delves into potential explanations while also acknowledging the limitations of classical MD simulations. They can't capture everything, especially potential chemical changes. However, based on what they can observe in the simulations, they suggest several factors likely contribute to this lasting effect.
Okay, like,
One major factor is likely the thorough disruption of the interpeptide hydrogen bond network that held the original anti-parallel aggregate together so tightly. Once these specific H-bonds are broken by the oscillating field and the peptides are dispersed or perhaps loosely reconfigured into different arrangements, those specific bonds required for the tight anti-parallel packing don't easily reform in the correct way. The system loses its memory of that structure.
so the specific physical connections are effectively scrambled and lost.
Yes, that's a good way to put it. They also suggest that steric clashes might play a role.
Steric clashes, like atoms bumping into each other.
Exactly. When the peptides are forced into these more disordered random coil states, or perhaps even into those loose, transient, parallel arrangements favored by the field, the bulky side chains of the different amino acids might physically obstruct each other. They get in each other's way, preventing the peptide backbones from efficiently rearranging back into the very specific, tightly packed structure required for the stable anti-parallel beta sheet aggregate. It's like trying to put puzzle pieces back together when they're bent or have stuff stuck to them.
Interesting. So physical blockage prevents reassembly. Are there other potential factors they mentioned that could contribute to this irreversibility?
They do speculate about the possibility of chemical changes contributing, even though their classical MD method doesn't explicitly model chemical reactions like bond breaking or
When
forming.
did that happen?
Well, processes like proton transfers, protons hopping between different acidic or basic groups on the peptides, are known to occur readily in aqueous solutions. It's possible the strong oscillating electric field could influence the rates or locations of these proton transfers. And if that happens, it could subtly alter the peptide's chemical properties, like their charge state or hydrogen bonding potential, in a way that makes reaggregation much more difficult, perhaps permanently.
So the field might induce subtle but lasting chemical modifications?
It's a possibility they raise. They also highlight an interesting comparison they did, shown in figure S12, where they simulated the neutral form of these peptides. Turns out the neutral form doesn't self-aggregate over long simulation times. This implies the zwitterionic form, the one that has separated positive and negative charges and is the stable form in water, is essential for the initial aggregation process. And perhaps how it behaves after disruption by the field might also depend heavily on maintaining that zwitterionic state or how the field interacts with it.
That's a subtle but potentially important point about how the peptide's specific charge state affects its aggregation and disaggregation behavior.
It is. And one final factor relates back to the energetics we discussed earlier. Remember how parallel beta sheets are generally energetically higher, less stable than antiparallel ones in the absence of a field.
Right, antiparallel is the preferred low-energy state on its own.
So while the oscillating field might temporarily stabilize or favor loose parallel-like arrangements, once the field is removed, the system doesn't just snap back to the lowest energy antiparallel form. It seems that pathway is kinetically hindered or structurally unfavorable after the extensive disruption caused by the oscillating field. The system gets trapped in the disordered state.
It's like the oscillating field didn't just temporarily push them apart. It effectively scrambled or rearranged them into a state that simply can't easily find its way back down the energy landscape to the original harmful configuration, at least not within the simulated microsecond timescale.
That seems to be the most plausible conclusion from the simulation data. It suggests the oscillating OEEF pushes the system over an energy barrier or into a different basin of attraction on the energy landscape, a disordered state from which the pathway back to the specific stable antiparallel aggregate is very inefficient, very slow, or perhaps completely blocked within realistic timescales.
Okay.
So yes, the combination of the rapid breakdown efficiency and this simulated irreversibility, likely due to a combination of disrupted interactions, steric hindrance, maybe subtle chemical changes, and the altered energetic landscape. That's really the core exciting finding here.
This simulation result has incredibly exciting potential implications, but as you stressed, it's rooted in computational work at this stage. How did the researchers try to relate these findings to existing medical techniques or therapies that are already out there?
That's important context. They put their simulation results into perspective by comparing them, at least conceptually, with two established therapeutic approaches that also use electrical or electromagnetic fields, albeit in different ways.
Okay, which ones?
First, they mention TT fields, therapy tumor treating fields. This is a non-invasive technique that uses alternating electric fields, but typically at much lower frequencies than the ones in their study. TT fields are usually in the 100, 500 kilohertz range.
Kilohertz compared to the gigahertz they simulated. Much lower.
Much lower frequency, yes.
Yeah.
And TT fields are primarily used to treat certain types of cancer, like glioblastoma. The idea there is that the fields disrupt the process of cell division in rapidly dividing cancer cells. So the similarity to this study is really just the non-invasive application of electric fields, even though the frequency, the target, cancer cells versus protein aggregates, and the likely mechanism are quite different.
Okay, so a conceptual similarity in using fields non-invasively, but very different specifics. What about the other comparison?
The other technique they compare to is deep brain stimulation, or DBS. This is a well-known invasive technique used for neurological disorders, most notably Parkinson's disease or essential tremor.
Right, DBS involves surgically implanting electrodes deep into specific brain regions.
Exactly. And these implanted electrodes deliver electrical impulses directly to the brain tissue. The parameters are again different, typically short pulses, maybe 60 microseconds long, delivered at a relatively low frequency around 130 Hz. Not the continuous wave gigahertz fields of the simulation.
Okay, so DBS is invasive, different parameters. How do the study's findings compare directly to the characteristics of DBS treatment?
The key characteristic of DBS they highlight for comparison is its typical usage pattern. DBS often requires continuous or regular application to be effective. Patients usually need the stimulation to be turned on to manage their symptoms, like tremor. And if the stimulation is turned off, the symptoms often return relatively quickly.
Okay, so DBS often needs to be constantly active because the underlying issue persists. Now, let's correlate that with the amyloid dynamics in their simulation again.
Right, so in their simulation model, spontaneous beta-amyloid aggregation formed in about 1 microsecond. The oscillating OEEF broke down that aggregate extremely rapidly in just 50 nanoseconds, about 20 times faster than it formed.
Got it. Fast breakdown.
But the crucial point of comparison with DBS is the simulated irreversibility we just discussed. The disrupted state, at least over the 1.5 microsecond simulation time frame after the field was removed, did not revert back to the aggregate. It stayed broken down.
So unlike DBS, where continuous stimulation is often needed because the underlying problem, like abnormal brain circuit activity, recurs quickly, the oscillating OEEF simulation suggests a rapid breakdown into a state that doesn't immediately revert. This implies you might not need continuous treatment with this approach.
That's the powerful contrast this study highlights, based purely on their simulation data, of course. Given the simulated speed of breakdown relative to formation, and perhaps more importantly, the simulated irreversibility or long-lasting effect, the authors propose that a periodic application of an optimized oscillating OEEF technique could potentially serve as a very promising therapeutic treatment strategy for Alzheimer's and maybe other related neurodegenerative diseases involving protein aggregation.
Periodic, not continuous.
Exactly. The simulation indicates it might not demand the constant application often associated with DBS for managing symptoms. It might be more like a treatment you get intermittently to clear out aggregates before they can significantly reform.
Okay, so let's try to bring this all together now. What are the absolute key takeaways from this deep dive into this really fascinating molecular dynamics study?
All right. From this rigorous computational work, we've learned several crucial insights, at least within their model system. First, these beta-amyloid peptides, represented by the KLVFAE segment, have a strong inherent tendency to spontaneously aggregate into the harmful antiparallel beta sheets that are thought to seed plaques. This is driven fundamentally by their large dipole moments and hydrogen bonding.
Right, that's the baseline problem. And we learned that static external electric fields can interfere with this. They can prevent spontaneous aggregation by favoring a different, less stable parallel structural arrangement.
But crucially, static fields aren't very efficient at breaking down plaques that have already formed. That breakdown process, in the simulation, takes roughly as long as the original formation, suggesting a need for continuous, potentially problematic application.
Okay. Then came the breakthrough finding with the oscillating electric fields.
Yes. Using the same moderate field strength, but oscillating it at microwave frequencies, specifically in the gigahertz range, the researchers observed a dramatically faster breakdown of existing aggregates, about 20 times faster than spontaneous formation in their simulations. Much more efficient.
And perhaps the most exciting result, the one that really points towards a potential new therapeutic strategy down the road, is that this oscillating field-induced disintegration appeared irreversible, or at least very long-lasting in their simulations. The peptides didn't spontaneously reaggregate back into the problematic antiparallel form even long after the field was turned off.
That combination of speed, rapid breakdown, and simulated irreversibility, lasting effect, is what makes this non-invasive oscillating OEEF approach seem potentially superior to static fields, based purely on these atomic-level simulations, of course. It offers a compelling new direction for exploring physics-based therapies for AD.
And as you said, it provides that favorable conceptual contrast with the need for continuous application often seen in techniques like DBS, suggesting a future treatment that might, hypothetically, only be needed periodically.
Exactly. Now, it's vital to reiterate this is a computational study. It's based on a simplified model, peptide, and simulations. Translating this to real biological systems, let alone human patients, involves enormous hurdles and requires extensive experimental validation.
Absolutely. Simulation is a starting point, not the finish line.
Precisely. But this study, while purely computational, certainly opens up exciting possibilities and provides a strong theoretical rationale for further investigation. It might encourage experimentalists to explore these kinds of oscillating fields. And it also echoes some really fundamental questions that the authors themselves raise right at the very end of their paper, prompting us, and you listening, to think more broadly.
Oh, what fundamental questions are those?
Well, they pose two big ones. First, they ask, is the beta-amyloid plaque truly the sole root cause of Alzheimer's disease? This is still a subject of intense debate in the field.
Right, the amyloid hypothesis isn't universally accepted as the complete story.
Exactly. Or, they ask, if a technique like this oscillating field were successful in clearing plaques in a biological system, could the observed clinical benefit actually be due to something slightly different? Specifically, could the benefit come simply from increasing the amount of properly folded, healthy, functional amyloid precursor protein or its fragments in the brain, rather than just removing the toxic aggregate?
Ah, that's a significant distinction, isn't it? Is the therapy working just by cleaning up the toxic mess, the plaques, or is it working by helping the brain restore its pool of normal, healthy proteins that were previously trapped or depleted by the aggregation process? It really changes how you think about the underlying mechanism of the potential therapy.
It does. It leaves us with this important, unresolved question about the precise relationship between plaque formation, protein misfolding, loss of function, and the downstream consequences that lead to the clinical symptoms of Alzheimer's disease. It's complex.
Definitely food for thought.
And the authors also briefly mentioned the possibility of applying this same kind of research approach using computational modeling to explore how electric field might target protein misfolding and aggregation to other proteins implicated in different neurodegenerative diseases. They specifically mentioned alpha-synuclein, which is the protein that aggregates in Parkinson's disease.
Right, so this isn't necessarily just about amyloid and Alzheimer's. Could physical methods like these tailored electric fields potentially be a broader tool, a platform technology for tackling protein misfolding across a whole spectrum of these debilitating neurodegenerative diseases?
It's a fascinating and potentially very impactful possibility that these simulation results put on the table for future research to explore. Could physics offer new weapons against these biological malfunctions?
Well, you have just taken a deep dive with us today into the incredibly intricate molecular world of beta-amyloid plaques and the tantalizing potential shown through this cutting-edge simulation work of oscillating electric fields to rapidly and perhaps irreversibly disrupt them.
We really hope this deep dive helps you appreciate the complex molecular battles that are happening within neurodegenerative diseases like Alzheimer's, and maybe spark some thought about the truly creative ways scientists are exploring physics-based methods using forces like electric fields to intervene at the most fundamental level.
It makes you think differently about those spontaneous forces pulling proteins together into harmful clumps, and the possibility of using carefully tuned external forces to push them apart again, maybe, just maybe, for good.
It's a compelling direction for future science.
Thank you so much for joining us for this deep dive.